informed. But for the moment, we want to be able to finish the semester, and that's good. And so us as a faculty member, we need to be a little bit more flexible. We say that we will receive a cut in pay to 20% if that will help the university. And they say no, they want to cut off faculty. So we'll see how the 2.0 version of this university will how it looks. Okay, so now I have a little bit more of people in the room, 46. I started with 20, so that's good. And we are going then into our, where is it? Okay, here we go. And now we need to put you guys in here so I can see. There we are. Okay, so this is the last image we saw on Monday. We were looking at this anterior-posterior pattern in mammals. And we saw the effects of, okay, here we go. WNT, BMP, and FGA to have a greater concentration at the posterior part of the embryo. And then we have the antagonist in the anterior part. So we have right there a gradient that will allow this part. And retinoic acid also playing a role in the posterior part of the embryo, activating some genes that determine the posteriorizing section of that embryo. And then we have that anterior-posterior. Now, once gastrulation begins, the anterior-posterior polarity of all vertebrates becomes specified, and that is made through the expression of the Hox genes. We have already talked about these genes that are playing a role and being the same type of genes in all individuals, right? Because we look at it in Drosophila, but we also see it in vertebrates, okay? So these genes, because of that reason, are said to be homeotic genes, right? So is the home C that we see in Drosophila. The home C genes are arranged in the same general order as the expression pattern along that anterior-posterior axis. And this the same way in flies as we will be seeing in mammals. Now, the order of the genes on the respective chromosomes are remarkably similar. And in addition to that, the expression of these genes follow the same pattern and are expressed anterior to the homologous in Drosophila. So we want to have, those are expressed anteriorly, are also expressed anteriorly in mammals. So genes that control the development of the head of Drosophila have homologous that control the development of the brain in mammals. That's what I want to say. So, and this is what we see in this image over here. So this is the evolutionary conservation of the homeotic gene organization in vertebrates. And we have the Drosophila and the mouse underneath in the image. So, and we have this anterior and posterior part. So here in this section of the figure, we see the similarity between the home C cluster in Drosophila, and this is on the chromosome 3. So just this section over here. And then we have the 4 Hox genes clusters in mouse chromosomes. So we have 1, 2, 3, and 4. Okay, so these are 4 different chromosomes, the Hox A, B, C, and D. Okay. And then the shade regions show particularly strong structural similarities between the species. And you can see that the order of the gene on the chromosome has been conserved. Okay, so we have exactly the same positions for some in here. The whole thing changes a little bit, but then we have almost the same. It's pretty well preserved across these species. Okay, so genes having similar structures, same relative position on each of the 4 chromosomes, and the similar expression pattern belong to paralogous groups. Okay, so we can compare this and look at duplications or replications, and they create more or less the same structures in both individuals. Now, because this is preserved across the species, we're going to talk about the Hox code. So the Hox code model predicts that combination of Hox genes specify the development of the anterior-posterior axis, as we saw in the previous slide. So it appears that there is a code of differing Hox gene expression along this anterior-posterior axis, and this code determines the type of vertebra formed by the individual. Okay. So evidence from this comes from 3 lines of experimentations that we have here in the screen. The first one is the comparative anatomy, in which the types of vertebra in different vertebrates is correlated with the constellation of the Hox gene expression. So which type of gene is expressed, and we're going to see that in the next slide. Okay. The second one is through gene targeting or gene knockout experiments, in which mice are constructed that lack both copies of one or more genes of Hox. And we know that without certain Hox genes, some regionally specification organs along that anterior-posterior axis will fail to form because it doesn't have the information. Okay. Sometimes it will become really specified in other regions if we put it somewhere else. So different sets of Hox genes are necessary for the specification of any region in this anterior-posterior axis. And maybe I'm going to make myself a little more clear once we get into the next slide with the color codes. And the third postulate, or the third evidence for this experimentation, is the retinoic acid. So retinoic acid teratogenesis, in which mouse embryos are exposed to retinoic acid, show an atypical pattern of Hox gene expression along that anterior-posterior axis. So here we go. So we have these images. We have the chick, and then we have the mouse. Okay, so we know that these are the points of expression of the different genes: 5, 6, 9, 12, 5, 6, 9, 10. I say 12. What I'm saying, I'm just looking at the 12 here, the Hox 10 over here. Okay. Now, if we know chicks, they have a different number of vertebrae than mouse at the different sections. So we have the cervical, thoracic, and lumbar, sacral, and coccygeal. Chick have 14 cervical vertebrae, while mouse has 7. Chick has 7 thoracic, mouse have 13, lumbar 12, 6, and then we have the sacral, we have 4 in there, and the caudal, because of the long tail, right, are 20. So we have a different number, but the point at which we are determining this is depend on the expression of the genes. Okay, so we know that the cervical to thoracic is in the transition between the 5 and the 6 gene expression of Hox. Okay. And then the thoracic and lumbar is at the level of expression of the 9 and 10. And we see exactly the same thing in mouse. So the position varies a little bit, but the point in which we change from cervical to thoracic, and we know that vertebrae are different. The cervical vertebrae are different than the thoracic, and the thoracic are different than the lumbar, right? So we see that expression depends on the expression. The variation of vertebra depends on the expression of these genes. And you see here, the right at the point, you have half and half. So we have a combination. So in both cases, HOX5, for example, parallels are expressed in the last cervical, while the anterior boundary of HOX6 parallels extends to the first thoracic. Okay, so that's the section over here. The last point that they had, the third point, was the retinoic acid. We know the retinoic acid induced homeostasis during mouse embryogenesis. We saw it a lot more expression of the back. So retinoic acid, we have a development, it's a developmental steroid and teratogenic agent too, and has been found to alter the actual expression of homeotic genes when administered during somatogenesis, somite formation period. Okay, so we know that retinoic acid causes the anterior expression boundaries of several anterior HOX genes to be shifted posteriorly, okay, in the formation of the vertebra. So the vertebra shifts anteriorly. What we have here in A, this is the wild type. So we have here the thoracic 13, and we see this type of ribs in this section, and then that's the thoracic, and this is the lumbar, and this is the sacral. So different type of vertebra, right? In B is a complete knockout of HOX10. So if I go back here is our 10, where we have this transition to lumbar, right? So what we have in here is the complete knockout of the HOX10 parallels that convert the lumbar one, this is the lumbar, into thoracic ribs, because this is the end, right? So we have more, it just had a knockout, and we can have, and then sacral becomes more like thoracic, okay? Because we have moved it. In C, it's a complete knockout of the HOX11. If we come back, HOX11 is going to be somewhere over here, the expression, okay? So in this case, because we have a complete knockout of HOX10, parallels transforms the sacral vertebra into copies of the lumbar vertebra. So we don't have them anymore, and then the whole transition occurs. So for the dorsal ventral axis formation.
Yes.
Ellen, you have a question?
Yeah, about the C. I don't understand the exact difference between C and the wild type. Like, I understand that the gene was knocked out, but I can't spot the differences.
Okay, so because it just, oops, the other way around. The knockout of 11 is somewhere over here at the very, very end. Okay, so all of those don't have, I'm going the other way around here. So you don't see it simply because this part of here is sacral, okay, but don't have these little things that normally is on the sacral part. They are more like thoracic to this end over here, but they should appear more like this end here that is sacral. So it's moved backwards. But because it's 11 don't determine thoracic. So this is why we don't have the expression like we did in this case here. That make this a little more clear for you? Okay. Okay, so for this dorsoventral axis, we don't know too much about it, okay? We know, well, particularly in mammals, of course. So we know that dorsal axis formed from the ICM, so the inner cell mass that we discussed last time, that are in contact with the trophoblasts. Remember, the trophoblasts are the external cells that are surrounding, okay? But also, this will result into the abembryonic axis. This is something that we're going to see in the lab of the blastocyst. So The embryonic region that contains the inner cell mass, the abembryonic region, is the part of the blastocyst that is opposite to the ICM. Okay. The placement of this ICM, the inner cell mass, appears to be specified by the point of entry of the sperm, almost in a similar manner as we saw for the sea urchin, remember? Or the frog, excuse me, where the complete opposite, we're going to have the great crescent. So the entrance, 180 degrees opposite to the entrance of the sperm, we're going to have the great crescent appear, right? So in this case, it's the same. It appears that it's the opposite direction where the sperm penetrates, where we're going to have that inner cell mass formation. So this dorsoventral axis of the embryo appears to form at the right angles to the animal vegetal axis of the egg. And then we have the left and right axis. We know that mammalian are not symmetrical, okay? Although the main gut, heart, and liver start almost in the initial position, central. This is 5 weeks of development. By 7 weeks, we see that the liver normally goes to the right, and then we have the heart that is going a little bit towards the left, and then the spleen and the pancreas also are moving, and this is just within 5 to 6 weeks, okay? So the left-right axis seems to be controlled through the activation of that gene that we have seen before, which is nodal, okay? And with the contribution, of course, of the protein, the PTX2, okay? Which is a transcription factor, when normally is expressed on the left side of the lateral plate. Okay, so we saw it in fish, right? We saw it also in frogs. So this is very conserved through vertebrates. Cerberus, we know, is normally expressed on the right side of that embryo, and that is an inhibitor of nodal protein. Okay, so once again, so the distinction between the left and the right side begins in the ciliary cells of the node. Remember, the node was how cells are gaining the inside of the embryo through the node. Okay, and that cilia are determined at the beginning how this goes. So in an experiment that caused a knockout out of the mouse gene coding for the motor proteins of the cilia at the node level, so the cilia will not move. The result of this lateral portion was randomized. It wasn't really organized. It was not. So even if we start with the heart, for instance, as a joining tube, we're going to see this in detail. By week 6, we start turning, coiling, and it gets a little more towards the left side. And the same, the only exception is mainly the lungs. We start like a tube, then start branching, and then we have the left and right. But even then, we have 3 lobes on the right and only 2 lobes on the left. So it's not symmetrical still. So, as indicated early, the early cells of the mammalian embryo can replace each other and compensate for missing cells, which means that these cells are pluripotent, right? And this is how we can do, when we do in vitro fertilization, normally we are going to take, to make an analysis of the embryo before we do the implantation. Parents that are going through in vitro fertilization, they want to be sure that their chance to have the perfect baby are on their side. So normally they're going to take one of the cells when they are in the more or less stage before doing the implantation to make some analysis for the baby, for trisomia, for any other diseases that can occur, okay? However, we know that human twins are classified into 2 major groups. we have the homozygotic, and we have the dizygotic. In the homozygotic, one egg or identical twins were formed from a single embryo cell that somehow divided, right? This homozygotic twins normally occurs 1 in 400 cases in humans. Now, the dizygotic twin, we have 2 eggs. So these are fraternal twins. And we have 2 fertilization phenomena occurring, okay? So homozygotic, they're identical twins. Dizygotic, where 2 eggs fertilize. So now, in identical twins, we can have some variation based on the timing for the formation. And this is what we have here in the screen. These are homozygotic twins, okay? Normally, we're gonna see 30% of the twin cases, okay? And go through the line A, this one, okay? When we're gonna have separation, normally occur before the formation of the trophoblast, okay? How we know that? We know already that the trophoblast is the one that forms the chorion, right? So in here we have 2 cells, and then they divide, and each of them have their own trophoblast, and each of them have their own chorion, okay? So 30% of the twinning occurs in this way, that before we have the blastocyst, they have divided, and each of them have the inner cell mass, and they have their own trophoblast because they have their own chorion formation. In B, what we have in here is some may share a common chorion, as we see it right here, okay, which indicates that the separation occurred after the trophoblast was formed, but before the amnion form. So each embryo finished with their own amniotic sac, as we see here. Each of them are in their own amniotic sac, but they both share the same chorion, okay? So this indicates that the split occurred at the level of the inner cell mass, right? So this is what we have in here, because they have their own amnion, but one chorion. So the division only happened there in the inner cell mass. The third way that how we can have this monozygotic twinning is when a splitting occurs after the amnion formation. So we have here our trophoblast, our inner cell mass, okay? And then something happened here, and then we finish twins in the same amniotic sac and in a single chorion. So all identical twins are not don't go necessarily through the same process, okay? Now I wanted to show you a video, but I need to stop sharing my screen for 2 seconds so I can go to the website. If I can find it. No, I didn't have time to do this between lectures. Okay, so it's here. Just give me time to share again. Oh, no. Now I can share. Which screen I share?
We can see the video.
You can see the video?
Yes, it's not
Yeah.
playing, but we can see it on Chrome.
Yeah, it's that I don't see it. Okay, here we go. Because you are on my other screen, but this other screen is not showing what I want to show.
Ever since we became cognizant of our births, twins are usually cataloged into three major types. There are fraternal twins, identical twins, and conjoined twins. Fraternal twins are often called two-egg twins or dizygotic twins because they are the product of two fertilization events. Identical twins are often called one-egg or monozygotic twins because they are the result of only one fertilization event. Conjoined twins, sometimes called Siamese twins, as we'll see later, probably occur after both fertilization and cleavage events have occurred. Now, to understand twinning, one really has to review some of the basic principles of mammalian embryology. Fertilization takes place in the oviduct. When the egg is ovulated, it begins the resumption of its first meiotic cleavage, but it arrests during the metaphase of its second meiotic division. Once the sperm enters the egg, the calcium ions that are released from within the egg enable the egg to complete its second meiotic division. The pronucleus that is formed then can migrate and meet the male sperm pronucleus, and when they get together, they start the first mitotic division. So fertilization takes place in the oviduct, and now the cleavage divisions start taking place. Cleavage in the mammalian egg causes the formation of a loose assortment of cells. Around the 8th cell stage, the cell membranes change, causing compaction to occur. When these cells divide, there will be a 16-cell stage called the morula, which contains a small group of cells on the inside and a larger group of cells on the outside. The inner cells will become the embryo, as well as the embryo's amnion and yolk sac. The outer cells become the trophectoderm, the embryonic portion of the placenta. Fluid will fill the middle space, causing the embryo to form what's called a blastocyst. The blastocyst has structure. The outer cells are the trophectoderm, sometimes called the trophoblast, the cells that will form the placenta. The inner cells are the embryonic stem cells. The embryonic stem cells will delaminate to form the amnion. This will fill with amniotic fluid. The trophoblast is incredibly important at this time because it constitutes the group of cells that will adhere to the uterus and will actually enter into the uterine wall. So at day 10 to 11, the embryo enters the uterus. It consists of the embryonic epiblast, which is to make the embryo, the amnion, which is to make the water sac, the yolk sac, sometimes called the visceral endoderm, which is important in coordinating the differentiation of blood cells and blood vessels, and it also gives structure to the embryo, forming the anterior-posterior axis of the embryo, and the trophoblast layers. The trophoblast has its single cells, the cytotrophoblast, and its multicellular syncytiotrophoblast. At about day 12, gastrulation occurs. You form the node, you form the primitive groove and the primitive streak. Cells will migrate from the epiblast through the primitive groove to form the mesoderm and the endoderm. Most of the hypoblast cells, most of the cells of the visceral endoderm are replaced by the cells migrating that forms the endoderm. The cells that will form between the endoderm and the epiblast will become the mesoderm. The cells remaining on the epiblast will become the ectoderm. Now, back to twinning. First, let's talk about fraternal twins. Fraternal twins do not have to be the same sex. They look alike or as unlike as regular brothers and sisters. This is because fraternal twins are the product of two fertilization events. So you have two sperms, two eggs. Sex determination could be different or the same in those two fertilization events. They form two separate embryos, two placentas, and they will form two newborns who are as alike or unalike as brothers or sisters. Now, this is different from identical twin or monozygotic twin formation. Monozygotic twins, single-egg twins, occur once in about every 400 births. About 1/3 of the identical twins have separate placentas, separate chorions. This means that the split occurs early in development before the formation of the trophoblast on day 5. So this is the earliest group of identical twins. Most other identical twins, about 66%, about 2/3 of identical twins, have a single placenta, but two amnia. They have two water sacs. This indicates that twinning occurred after day 5, the formation of the placenta, of the trophoblast, but before day 9, when the amnion forms. Only around 1% of identical twins have a single amnion and a single placenta. This means that twinning occurred after the amnion forms on day 9. This shows regulation and pluripotency, two very important parts of mammalian development. Pluripotency is the ability of a cell to become any cell in the embryo. Regulation is the ability of a cell to become what it does, to have a new fate, depending on where it is in the embryo. So if twinning occurs, you don't get two halves of an embryo. You don't get a right half and a left half or an anterior and a posterior. You get two fully developed newborns. Now, the opposite of twinning is chimerism.
Okay, I'm just gonna stop at 2 seconds so I can give that part. And I'm sorry, Robert, I couldn't let you in because if I click to let you in, the video stops. So.
Yeah, no worry. I got disconnected.
Okay, sorry. Okay, so now let's go to here. Okay, so that's how twins can be created. Now, while fraternal twins are formed during fertilization of two separate eggs, identical twins, as we saw and explained, are formed during cleavage, but conjoined twins are created during gastrulation. Okay, so two timings are completely different. So in here we have conjoined twins are often of the same sex. Okay, so it occurs 1 in 200,000 live births. We're gonna have a conjoined twin. So in figure A over here, what we have is that this is a chicken. In the chicken, for instance, so we have the blastodisc, right? And the epiblast is divided in 4 parts. And each part will form a parameter streak. And then we're gonna have 4 individuals, okay? In figure B, in here, we observe what happens if a tear in the marginal zone, which normally, remember, marginal zone expresses nodal, okay? So we're gonna have a second axis that's going to form. So here we have, and this is the second neural tube, and this is the second head forming, and this is the first one. This is the original, but because it was a tear at the level, we know what the nodal is, then we have a second axis forming. In C, shows what could explain the occurrence of conjoined twins, where 2 areas of the margin producing nodal are present. So we have, depending on where they occur, we have craniopagus. The embryo is going to be jointed by the head. Pygopagus, they're going to be joined at the back, lower back. And then we have parapagus, is lateral joining. And then we have thoracopagus, that is in the thorax. So depending on where that second axis occur, then they're going to be joined. Okay. Okay, so this is going to bring us where we were before, but now we can just skip to the minute wherever we were on this other one. Oh. And then I'm gonna share again the other screen so I don't need to go back to. There we go. Oops, what happened? Oh. This one.
Here is when you take two separate embryos, as in fraternal twins, you have two fertilization events, and you fuse them together to make a single embryo. So you could take mice, which are the product of white furred parents or the product of black furred parents. You take these two embryos, one which is going to have white furred mice and one which will result in black furred mice, and you mix them together. You mix the cells together. And what you get is a chimeric blastocyst. And you put that chimeric blastocyst into a mother mouse who's been hormonally changed so that she's ready to accept this embryo. And what results is a striped mice, a mouse with areas of white fur and areas of black fur. It was hypothesized that you needed 3 cells in the inner cell mass to make an embryo of a mouse. So Clement Markert and his colleagues decided to see if they could make a chimeric embryo with 3 embryonic components. There was an embryo from white furred parents, an embryo from black furred parents, and an embryo from orange furred parents. These 3 embryos were mixed together. They put this embryo, this chimeric embryo, into a foster mother and waited for it to be born. And when the newborn came out, it was pink, because all mice are born without hair. They're all pink. So they had to wait to see what happened. And what happened was three cells could form the embryo. They wanted to see if the embryo had germ cells that were derived from each of those 3 embryos. And to find this out, they mated this female mouse with a white furred male, a recessive male. The offspring turned out to be white furred, brown furred, and black furred. In other words, all 3 embryos contributed germ cells to this hexaparental 6-parent mouse. We have fraternal twins, we have identical twins. Now, conjoined twins present us a mystery. We don't really know how conjoined twins are formed, but one very interesting theory is that conjoined twins are formed during gastrulation. It's been found in chick embryos that any spot along the edge of the epiblast can give rise to a new primitive streak. So the ability to form a primitive streak, the ability to start gastrulation, actually exists throughout the embryo. But once a particular spot is fated to become the site of gastrulation initiation, it inhibits the other sites. So we have here an experiment that was done by Spratt and Haas back in the 60s, where they actually divided the chick epiblast into 4 parts, each of which formed its own primitive streak. It's been shown in frogs and in chicken embryos that if you add an ectopic organizer or an ectopic node, you will get two new embryonic axes, two dorsal ventral, two normal axes will be forming, and that they often fuse together, as in conjoined twins. So Levin and others have speculated that the different types of conjoined twins depend on where a new axis is formed during gastrulation. So if it's formed in the anterior, you get twinning such as craniopagus twins, where the twins are joined at the head. If it is lateral to the original primitive streak, you get parapagus twins, which have two embryonic axes starting from a common point. Sometimes, as in thoracopagus twins, which are twins that are joined at the thorax, you will get a lateral organizer coming to fuse with the original organizer. And in piciopagus twins, that's when they are joined at the posterior of the embryo. And if an organizer starts there, it might join with the original primitive streak in development towards the posterior of the embryo. So twinning, the different types of twins, can be explained as events occurring during different stages of mammalian development. Fraternal twins, two-egg twins, arise during fertilization. Monozygotic, single-egg, identical twins arise during embryonic cleavage, and conjoined twins probably arise during gastrulation.
Okay, so I hope that video helped to understand a little bit. It's a mystery, right? How these things happen. For instance, in a vertebrate, the armadillo, the knife-banded armadillo, they do have quadruplets. Every pregnancy is a quadruplet. And this is how that happens. So they all have the same sex, and they're all twins. It's just a mystery of nature. Okay, so these two last slides are just to sum up a little bit what we cover in this chapter 12 and 11 and 10. So it's early development in the four classes, vertebrate classes. So cleavage differs greatly between the four groups. Fish and birds have meroblastic discalded cleavage. Okay, so here we have the zebrafish and the frogs. Frogs have unequal holoblastic cleavage due to the amount of vitellus that the eggs contain, right? On the other hand, in mouse, what we have is equal holoblastic cleavage. Okay, so these are the early cleavage and these are the late cleavage. These cleavage patterns form different structures, but each except the mammals has a nucleus center. We didn't saw a nucleus center in mammals, right? So, but all of the rest have a nucleus center that provides the signal to the organizer. We don't see that in mammals. Now, as gastrulation begins, okay, each of the groups had cells that are the equivalent to the organizer, or that what we call the Spearman-Mantle organizer, and that is the little red circle, right? In here, that's the dorsal lip, what happens, and in here, there, and even in mammals, even though we don't have the structure over here, the nucleus center, we do have this organizer, the Spearman Gold organizer. So this organizer marks the beginning of the blastopore region independently of the species. So that organizer always marks the beginning of the blastopore region, and the remainder of the blastopore is indicated. So you can see it in here, there, and then in here where the node will gonna be. As gastrula continues, where did my mouse go? Okay, here we go. As the gastrula continue and we start having that movements, all the different movements, invagination, delamination, epipole, conversion, extension, and all of those movements that you know by now, we know that the endoderm is going to be placed in the inside, right? And then the ectoderm is in the blue purplish color, okay, and surrounds the embryo, and that the mesoderm, which is always in red, is normally between the ectoderm and the endoderm. So the regionalization of the mesoderm has also begun depending on which part of this anterior-posterior axis it is found, okay? So to sum up, in frog, excuse me, you need to start with fish. In fish, the organizer is the embryonic shield, right? But the nucleus center is the yolk syncytial layer, okay, on the marginal and the embryonic shield, okay? In frog, okay. Then the next one is in frogs. So in frogs, the organizer is the dorsal lip blastopore, right? And the nucleus center is the dorsal-most vegetal cells under the marginal zone. In birds, we have the organizer is the Hensen's node, but the nucleus center is the posterior marginal zone, okay? And in mammals, the organizer is the node, and we don't have nucleus center. That's all, okay? So, oops. Here. I just wanted to finish this section on the mammalian. Normally, this is the time at which I normally finish. And I'm just going to cut it short today, the lecture. We're just going to leave it there. I don't want to start the next chapter that talks about, the lateral plate mesoderm. So the initial part for the formation of neurula, we just finished here, gastrulation. And I don't want to start the subject and to have a couple of slides and then continue on the Monday coming back from the reading week. So I prefer just to cut it short here. And we start on Monday coming back from the reading week. Okay. And if you have some midterms today or tomorrow, so maybe you can enjoy those 20 minutes either to relax and have a big breath and then continue your studies, or do whatever you feel that you gotta do, okay? I'm gonna see you tomorrow in the lab, okay? And the topics for the term paper coming back from the term, the reading week, okay? So you can get organized. I don't know if you have other work that you gotta do over reading week. Normally you do have some other midterms, so I don't want to overwhelm you with that. I'm trying to do my best to have your work not at a time that the hecticness of some other courses occur this semester. So I'm trying to be sympathetic to the cause of the students and this virtuality and midterms and all of that. And no, Robert, the topic's gonna be assigned by me. So I already have the topics. I just need to make a little bit more of research regarding some of them. But I'm gonna be picking up the subjects I have already 4 different subjects. I just need to be sure that you are going to be able to find sufficient literature on 2 of them. The others, I know that there is enough. But so as I do normally, I can assign topics independently. This is an individual work, okay? And you're gonna have until the end of the semester to do it. So as I say, this is just to practice different types of scientific communication, right? So one was the video, that is the oral one, and then is the written part. So 2 ways how scientific communication occurs. And depending on which way you are going next, I believe that you will be practicing both, either how to present yourself in a conference, and results into a board, into a patient. You're gonna need to transmit information. Or if you continue like in a master's or something, well, you're gonna need to write articles, you're gonna need to write a proposal. So this is all part of that training in scientific communication that I like to have in my courses. Okay, so having said that, I wish you a good afternoon, and see you tomorrow in the lab. Okay, bye-bye.